In this video, we'll give you a high-level introduction to Benchling molecular biology. Molecular biology is the core application in Benchling for your company's sequence design and analysis. Molecular biology offers more than 10 cutting-edge tools in one, including annotations, alignments, primer design, CRISPR guide design, and assembly wizards. As a Benchling for Startups client, you have access to bulk tools, including our bulk assembly wizard for modeling cloning and designing constructs in batches. Molecular biology offers a collaborative environment. Your team can track sequence edits and create shared libraries. For easy record keeping, your sequences are directly integrated with your notebook. Let's get started with the molecular biology application. First, we'll show you how to create and import DNA and protein sequences in Benchling. Then, we'll show you how to customize your workspace and how to navigate to each of the different tools in Benchling. We'll follow this video up with a separate molecular biology deep dive, which goes into specifics for several key features.
To create new DNA sequences, click Global Create, DNA Sequence, and then either New DNA Sequence or Import DNA Sequence. Each of these will open a different tab of the same menu. The menu gives you 4 options: converting files or uploading existing DNA files, searching external databases, selecting chromosomal regions, and inputting a raw sequence. From the Convert Files tab, you can upload existing DNA files in various formats to Benchling. Simply drag and drop your sequence from your desktop, or choose a file from your desktop to upload. From the Search External Databases tab, you can search AdGene, NCBI, Ensemble, and other external databases for sequences that you wish to import. For example, let's say we want the mouse sequence for BRCA1. We can search BRCA1 and then select the mus musculus genome from the dropdown. Customize the parameters in this menu as you need. From the Select Chromosomal Region tab, you can import a known region in a genome of interest. Search for your genome, select the chromosome number, and choose the start and end positions of the sequence. From the Input Raw Sequence tab, you can create a new blank sequence. When the new file opens, you'll be able to type or paste your sequence into the file directly. You can create new amino acid sequences from the Global Create menu as well. Under AA Sequence, click New AA Sequence or Import AA Sequence. If you click New AA Sequence, you'll have the option to create a new blank protein sequence file, just like you could create a blank DNA sequence. If you click Import AA Sequence, you'll have 2 options for importing sequences: uploading files and importing from databases. From the Upload Files tab, you can drag and drop or choose files to upload. Benchling handles FASTA, GenPept, and UniProt XML files. From the Import from Database tab, you can search the accession number of a protein sequence from NCBI or UniProt. You can also create new sequence files from existing sequence files. To do so, you must select a region from an existing sequence, like this DNA sequence. Drag your cursor over a specific region or click on an annotation to select the annotated region. Next, right-click the selected region to open a drop-down menu. From this menu, you can select Create DNA Sequence to create a new sequence file with the selected region only. You can also click Create AA Sequence to create a new protein sequence corresponding to the selected region. You must choose whether you want to use the forward or the reverse sequence. You can also annotate the given sequence with the corresponding protein sequence by selecting Create Translation. To open a sequence file, navigate to where it's stored and click on it. Once open, the tabs at the top of your workspace give you options for viewing your sequence and keeping notes about your sequence. To see multiple tabs at once, click the Split Workspace button at the bottom right-hand of your screen. At any point, you can click Split Workspace again to view only a single tab. In your Split Workspace, you can see your sequence map alongside a plasmid map. Keep any relevant notes about your sequence in the Description tab. This is especially helpful for sharing context about sequences with members of your organization. Drag the tabs around to customize your split workspace as you need. Our molecular biology application is full of rich features to help you design, analyze, and customize sequences. We'll show you how to navigate to all of these features, starting with the top navigation bar. You will see several buttons at the top right of your sequence map. The Create, Copy, and Blast buttons can be used after you've highlighted a specific region of your sequence. The Create button allows you to create annotations, create primers, translations, and new protein or DNA sequences from your selected region. If you click the Copy button, you can copy the sequence you've highlighted, its reverse complement, or its forward or reverse translation. You can paste any of these sequences into another sequence or into a blank sequence. The Blast button allows you to send your selected region to NCBI Blast directly from Benchling. To download a PDF version of your sequence, click the PDF button. The Gear button gives you several options for viewing features on top of your entire sequence, from annotations to cut sites to translations. Check or uncheck any of these boxes to focus on the features that you care about. Finally, the Search button opens up a search bar that allows you to search your entire sequence for any bases, annotations, or primers. The right navigation bar is where you can access the majority of the molecular biology tools, such as annotations, primer design, alignments, and CRISPR. We'll walk you through each of the buttons on this navigation bar, one by one. The top icon, a small flag, opens annotations. From the menu that opens, you can select or deselect annotations, add new annotations, and use our feature libraries that enable auto-annotation. The scissors icon opens digests. From this menu, you can choose parameters for simulating restriction enzyme digests. The icon with two arrows opens primers. Create new primers, link primers in a pair, export primer sequences, and attach primers to a DNA sequence. The clock icon opens your sequence version history, just like the version history of your notebook entries. Customize the names of different versions, reset your sequence to old versions, or clone a version into new DNA. The next icon opens alignments. From here, you can create a new alignment using your open DNA sequence as a template. The icon below opens CRISPR tools. Using these tools, quickly design and analyze guide RNAs and design homologous recombination templates. Finally, the eye icon opens additional customizable information about your sequence. Most notably, you can change the topology of your sequence from circular to linear, or vice versa, by opening the topology dropdown. The bottom navigation bar in molecular biology also offers a useful set of features. First, on the left, it shows you the number of bases in your sequence and the position of your cursor in the sequence. When you select a specific region or the entire sequence, the navigation bar automatically populates with properties of that selection, including its start and end positions, length, %GC content, and melting temperature. Finally, you can open the Assembly wizard from the bottom navigation bar. If you click Assembly Wizard, then Create New Assembly, you'll see options for performing digest and ligate, Gibson, and Golden Gate assemblies.
Thanks for taking this tour of Benchling Molecular Biology with us. Molecular biology is a powerful application with over 10 sequence design and analysis tools that are user-friendly and highly customizable. It will allow your company to standardize and collaborate on sequence data, saving you time and allowing you to carefully manage your data from day one. There's a lot more to learn to get the full value of the rich features of Benchling Molecular Biology. We recommend watching our final training video, Molecular Biology Deep Dive. In the deep dive, we'll show you in more detail how to use the tools like annotations, primers, alignments, and CRISPR.